Okay, perfect. Hopefully you guys can see that. Alright, so this is what you need to do when you're taking my test, and a lot of people are like, I don't know what to do when I'm taking your test. I'm like, don't tell me this on test 3 and not test 1. So, here's what you have to do, look for the keywords. You see where it says 5-carbon sugar, and you're looking for an alternative to traditional carb metabolism? 5-carbon would be pentose, so it's B. The most important products in the Krebs cycle. First up, oxygen is not going to be made here. Oxygen is actually made in the ETC, the electron transport chain. 36 ATP molecule, that's made in the electron transport chain. And DHAP and GP, where do you see that? In glycolysis, when we break glucose, which is 6-carbon, we split that in half. DHAP and GP would be the 3 and 3. And then the answer then would be the coenzymes, reduced coenzymes. You need to make reduced coenzymes in the Krebs cycle so it can go into the electron transport chain. The next thing, the synthesis of sugars using carbon dioxide and carbon gas is carbon fixation. What are you looking for? Carbon. So you want to fix carbon. So nitrogen fixation would not be called nitrogen gas fixation. It's just nitrogen. Okay, let's talk about holoenzymes. The protein portion is the protein is the apoenzyme, and then the non-protein would be the cofactor, but if the cofactor is organic, it is the coenzyme. What's the enzymatic RNA called? Ribozyme. So rhizopus is a type of fungus. A rhizoid would be pretty analogous to a root, but it's in a fungus. The ribozyme is the enzymatic RNA, and the ribosome is your organelle that's the site of protein synthesis. Now, oxidation is what? Loss of electrons. When you're losing electrons, you're breaking bonds. What are you doing in glycolysis? Breaking glucose. Glycolysis, glycolysis. Break the glucose. So aren't you oxidizing it? If oxidation is a loss of electrons, and you need to break the glucose, aren't you losing electrons? There you go. So glycolysis will be the oxidation of glucose into pyruvic acid. Factors influencing enzymatic activity, temperature, pH, both of those, if extreme, will denature the enzyme. Substrate concentration and inhibitors will slow down the speed of your enzyme. So all of them. Okay, whatever. What was enzymes end in? Ase, okay. If you take glycerol and a fatty acid and put them together, you get a lipid. It's not there. I like doing that to you. You know the answer and it's not there, and then you go crazy. The answer is none of the above because glycerol and fatty acids come together to make a lipid. Okay, this one. In phosphorylation, what's going to happen? The electrons will be excited. They will then jump to the carriers. Now the electrons can come back to the chlorophyll, or they can not. So if they don't come back to the chlorophyll, we call this non-cyclic. Cyclic is if those electrons return. In this case, it's non-cyclic, and the answer is no. This one's easy. Which one here says light? There we go. So if you're an autotroph, this is what you do. You take carbon dioxide and you can pull carbon from it. So that would be a autotroph. If you're a heterotroph, you need to get your carbon in other ways. So here you're saying you're using light, you have to get carbon some other way, So you're a photoheterotroph. Polysaccharide synthesis produces glycogen and peptidoglycan because they both have sugar. A and B. Again here, oxidation, loss of electrons, the oxidation number goes up. You will get more oxygen or something like it. And if you're oxidizing one thing, another thing has to be simultaneously reduced. Here, okay, you're gonna go from 3 to 5 prime. What this means, on your nucleotides, your ATCG, there's one end that has a hydroxyl group, it's an OH, right, oxygen and hydrogen. Another side has a phosphate group. And so your nucleotides will go hydroxyl phosphate, hydroxyl phosphate, hydroxyl phosphate, hydroxyl phosphate, hydroxyl phosphate. So you will have on one end hydroxyl, on one end phosphate. That's what we need 3 to 5. The 3 end is hydroxyl, the 5 end is a phosphate. So when you're transcribing DNA into RNA, you'll take a strand. I didn't tell you what strand, so here you need more information. But what you would do, you would split these strands, and if this is 3, add a 5. 3, add the 5. Add the 5, add the 5. So you read your sequence 3 to 5, but you will synthesize it the complementary way, which is 5 to 3. So where you have a hydroxyl end, you must add the phosphate end. That's the whole process. So RNA is synthesized 5 to 3. DNA is synthesized 5 to 3. You can read them 3 to 5. So DNA polymerase or RNA polymerase attaches to the 3 end and goes all the way to 5. But as it's doing that, it makes the other strand 5 to 3 along with it. So it's like if it makes its, I don't want to say mirror image, but yeah, I guess it is a mirror image. Okay, so what's an example of selective media? Mannitol. Blood is differential and solely differential. Mannitol is selective and differential. And nutrients, agar or broth, grow everything under the sun. This one, just look at your sequences, look at the sequence, match up the table, I believe you'll see. This one, I think it was none of the above because one letter was off. Anyways, the longest phase of bacterial growth? Lag. That's what it's called, lag, because it lags. An example of differential media would be? Blood and mannitol, so both of them. So I'm not, if I said which one there is strictly differential, blood. But I'm just saying which one's differential, blood and mannitol are both differential. Okay, so removing all forms of life would require a? If you have 1 million bacterial cells, how long should you kill it for? 7. Just add a 1 to the exponent. Okay, genetic makeup is the genotype. So think genotype, genes. You have a bunch of genes, you don't express everything. That's why I said there's potential properties. You don't want to express everything that's in your genome, trust me. Now, the RNA protein complex that removes introns and splices exons would be? SNRPs. Yeah, SNRPs. Non-ionization radiation is what dimer? Thymine dimer. Well, I put trimine in there just to be neat. And mess you guys up. So, chemical agents, all of them. Aldehyde, alcohol, phenol, halogen, they're all chemicals to control bacterial growth. What bacteria will lack enzymes to neutralize its harmful oxygen? Obligate anaerobes. Because if you're an obligate anaerobe, you're never really supposed to be in contact with oxygen. So if you're not going to be in contact with oxygen, why are you going to expend energy to make something to help you against oxygen? So obligates arrows, facultative anaerobes,